section 7.3, we're talking about how we get things across the selectively permeable membrane, the plasma membrane, but now this section is about how we do that without using any energy. So this is called passive transport. Passive transport is when we transfer things across the cell membrane using no energy. And a definition you should know is diffusion. And diffusion is the movement of molecules of any substance as they spread out evenly into a space. So basically, if you have a given amount of water molecules, or a given amount of solute molecules, let's say sugar molecules, if a given amount of sugar molecules, they're going to randomly bump into each other and spread out until they're evenly spread out across the space that they're given. And diffusion is totally random, like literally the molecules just bump into each other and move off. But they're going to tend to move from areas of high concentration to low concentration, so it's going to be directional, meaning they're going to move one way if there's a concentration difference between the two of them. And if you think back to what dynamic equilibrium is, I think we talked about this already. Dynamic equilibrium is when there's no net change, but things are moving both ways. So like a chemical reaction, it can go backwards and forwards. The things that are diffusing are moving both ways. So I just mentioned a concentration gradient. Let's see if there's a picture for that. Okay, so a concentration gradient is when there's a difference in concentration. So if you look at this picture right here, this solute right here is a dye. So this is like if I put a thing of food dye into a thing of water, and there's a lot of solute on this side of this semi-permeable membrane, okay? And there's not a lot of solute on this side. So we have high solute concentration to low solute concentration. So these solutes are going to bump into each other and spread out. So we see net movement. Even though it's random, they're still moving from the right to the left because they're moving down their concentration gradient. They want to spread out. It's like as if I packed all of you students onto one half of the classroom. So I'd move all your chairs to that half of the classroom. And by the end of the day, you guys would, you all move around your seats anyways, you would eventually start to spread out. And if I let you, you would probably move and spread out across the whole entire classroom because you don't want to be that close to each other. It's just like with the solute molecules. They're moving away from each other. Eventually, they're going to be equally spread out. We have the same number of solute molecules on this side of the membrane as we do on this side of the membrane. And they're still moving because they haven't stopped moving. But for every one that crosses this way, one is going to cross this way. So they're at dynamic equilibrium. Dynamic meaning it's still moving, but equilibrium the same amount on both sides. And this can happen with also two solutes, okay? So each solute only cares about its own concentration gradient. So this dye right here, the orange dye, only cares about its own concentration gradient. And this one right here only cares about its own concentration gradient. So even though there's more orange dye molecules than there are purple, that doesn't matter. The orange is still going to move right and the purple is still going to move left until they're both at dynamic equilibrium. So over here, now we look pretty spread out, except you can see there's still so many more purple over here than there are over here and more orange over here than over here. So they're still going to keep moving until they're both equally spread out across the selectively permeable membrane. And now they're both at dynamic equilibrium. So this is what it's like inside of every cell ever. So nothing's ever going to be totally at dynamic equilibrium because that would basically be a delta G of 0. We would all die. So we want this to happen both ways. So at some point, some solutes might be at dynamic equilibrium, whereas others, sometimes our body works to keep things having a concentration gradient because that's how important it is to our cellular processes. So like I said, substances diffuse down their concentration gradient, meaning they go from high concentration to low concentration. Gradient is a difference, so it's a concentration difference. By definition, the region along which the density of a chemical substance increases or decreases. So right here, this orange molecule is at a higher density on this side than it is on this side. This does not require any energy to move things from high concentration to low concentration. No energy, no delta G, no change in H, because they're just bumping into each other and spreading out. We're not putting in any energy at all. And like I said, we call this passive transport because we're not putting in any energy for it to happen. It's just passively happening. It just happens on its own. No big deal. Passive transport. One of a very specific kind of diffusion is osmosis. And water is the only kind of substance that has its own definition for diffusion, and that's what it is. Diffusion, or osmosis, is the diffusion of water molecules across a selectively permeable membrane. This is a little bit tricky to think about because water is never the solute, it's always the solvent, so it's a little bit backwards from what we were just talking about. So water will diffuse across a membrane as well, but it's going to go opposite of the solute. So it wants to help even out the solute concentration. So it's going to move from areas of low solute concentration to areas of high solute concentration to help that equal out the concentrations on both sides. So here we have a picture. So this is a U-shaped thing with a U-shaped tube with a selectively permeable membrane. And you can see that on the left side of the barrier, there's far fewer solute molecules than there are on the right side of the barrier. However, this selectively permeable barrier membrane does not allow sugar molecules to cross. Okay, so the pores in the selectively permeable membrane are so small that only water can fit and sugar molecules cannot. So these sugar molecules cannot go from right to left to cancel out the concentration gradient. They want to, but they can't. So they're whatever, they can't do it. So instead, water can cross. Water is going to move from areas of low solute concentration to areas of high solute concentration. Because now look at this. Now the concentration on both sides is about the same because there's more liquid over here. So for every amount of water, there's the same amount of solute on this side as there is on this side. So it's evened itself out. This will actually happen. So even though the water levels aren't even, the concentration gradients are the same. So water moves from areas of low solute concentration to areas of high solute concentration. That way it could even out the solute concentrations. Now this picture, this is something important to know. So the water that's moving is only the free water molecules. The only water that counts in a concentration is the free water molecules. And what that means is, remember that water is going to make a hydration shell around anything it dissolves. Because water is polar, is going to stick to any charged part of a solute. So all these waters that are stuck to these glucoses are called, they're not free water molecules. So only the free water molecules are going to be what is diffusing via osmosis. And now it's important to talk about what this means for life. So we have this word called tonicity, which is the ability of a surrounding solution to cause a cell to gain or lose water. So when you drop a cell into a solution, is that solution going to make the cell absorb water or get rid of water? And there's three kinds of solutions: isotonic, hypertonic, and hypotonic. Iso meaning the same, hyper meaning more, hypo meaning less. Iso, just have to remember that means the same. Hyper, think about it, if you're hyper, you have a lot of energy, so it's more. And hypo, like, that just means less. So an isotonic solution is a solution, it's this one in the middle, where the solute concentrations are the same on the inside and on the outside. So there's going to be no net movement into or out of the cell. So let's look at this red blood cell. I said no net movement. So there's still going to be water going in, there's still going to be water coming out, but there's no net movement. The size of the cell is unchanged. The solute concentrations are unchanged. No big deal. Moving in and out the same. Then we're going to talk first about the animal cell. If that solution is hypotonic, that means that there's a lower solute concentration outside of the cell than inside. This is like if a cell is in fresh water, and water is going to go into the cell because there's a higher solute concentration inside, and water moves from areas of low solute concentration to high solute concentration. So the water starts moving into the cell, and the cell starts filling up with water because water is leaving, but not as much as is coming in. And so now the cell starts filling up and filling up. It starts blowing up like a balloon. It's eventually going to swell, and it's going to lyse. That's what we call when a cell pops. So the cell is going to pop like a balloon that you blew up too much in a hypotonic solution. In a hypertonic solution, we can see that there's a higher solute concentration outside of the cell. So the water that's inside the cell is going to move from low concentration inside the cell to outside of the cell. This is like if you dropped a cell into salt water. This is why it's dangerous for you to drink salt water. Now all the water is going to leave your cells, and the cell's going to shrivel up and look like this. And that's not good. And that's what happens to red blood cells in those three solutions, or any animal cells. Any cell without a cell wall, that's what's going to happen. Plant cells are different because they have a cell wall, so it functions a little bit differently. Isotonic is the same. Water moves in, water moves out. The cell would appear from this to be unaffected. However, a lot of this water is stored inside the vacuole, and the vacuole is what keeps the cell really stiff, because plants don't have skeletons, so they need to be stiff and they need to stand up straight using their cell walls. And so if they are at an isotonic solution, they're going to be a little bit limp. This is when they might start to wilt a little bit. So at isotonic, the cell is what we call flaccid. That means limp. At a hypotonic solution, more water is moving in than out. The vacuole is very full, and the cell, all the water is pushing against the sides of the cell wall, and we call that turgid. It's very firm, and it's allowing the cell to stand up and be full. So that's what happens in a hypotonic solution for a cell with a wall. In a hypertonic solution, it's not good news for a cell with a wall. As you can see, in an isotonic, it's already not good. I know it's hard to see because my face is covering it. But in a hypertonic solution, what happens is all the water leaves, but it doesn't shrivel up because it has a cell wall, which is going to stay there. But what happens is the cell membrane starts to pull off the inside of the cell wall. So you can see here that it's no longer attached. This is called plasmolyzed. We just basically pulled the whole cell membrane off of the cell wall. So for an animal cell, isotonic is ideal. For a plant cell, hypotonic is ideal. And hypertonic or hypotonic solutions or environments create osmotic problems for organisms. This is what I was just saying. If you drink too much salt water, you're creating an osmotic problem for yourself, because now all the water is going to want to leave your cells and go to the salt water, because it's low concentration inside your cells and high concentration outside. So this is like what happens when if you eat a lot of salty chips, your lips get really dry. That's because there's all this salt on your lips, and all the water leaves to go cancel out the salt on your lips. So that's an osmotic problem. So the process by which you regulate your osmosis throughout your body is called osmoregulation. This is controlling solute concentrations in water ballasts, and it's a really important regulation for or adaptation for life, obviously. Some single-celled organisms, such as paramecium, they maintain a solute concentration because they live in a hypertonic solution, so their pond has more solutes than they do. And so what they do is they are hypotonic to the pond, so they live in fresh water, so they take on water a lot because the water is moving from low concentration outside to high concentration inside. And so what happens is they have this vacuole that they fill up with all the water that's rushing into their cell, and they just pump it out. So they have a mechanism to maintain their osmoregulation. So there it is. There's the contractile vacuole. We talked about all of this. Okay, talk about this in the next video.